have more mice per square kilometer than elephants. Okay, so then I selected virtual species using the Comadre and Compadre databases to define reasonable vital rates from empirical models. So this shows all the models from the Comadre and Compadre database that met. We did some filtering for wild and unmanipulated populations, removed populations that we thought had data issues, and then we collapsed all of them to the same 2x2 model with 5 vital rates that I'm using, and used a PCA to define a life history space occupied by those empirical MPMs from those 5 vital rates. And this is just a couple of example species that kind of sit on the edges of the space that we defined. So then I selected a high and a low value for each vital rate, except for reproductive output, which is not free to vary. And that gives me 16 virtual species. And then there are 5 density-dependent vital rate scenarios. And I projected those onto the PC axes. So you can see that we're filling out that life history space or that vital rate space pretty well with these virtual species. So then for all of those virtual species across the different density-dependent scenarios and different values of population density, I calculated resilience metrics. So this is now just the virtual species on those same PC axes. This is compensation, which is the propensity of a population to boom after a disturbance. And we see that it's highest in this upper part of the PC space and lowest over here. And that's roughly fast life histories have high compensation, and slow life histories have low compensation. We see something a bit opposite in resistance. Resistance is the ability of populations to resist further declines after a perturbation. That is most strongly related to juvenile survival in these models. So it's also higher in slow life histories. And then this is recovery time, which is the number of time steps required to go back to that asymptotic behavior, getting back to the stable distribution and growing at a rate lambda. And it has a little bit of a more complicated pattern, and we'll talk more about recovery time later. So, the last thing I wanted to say here is that these results are generally consistent with past results on density-independent models from Paul Capdevila, and where he found likewise that compensation was highest in fast life histories and resistance was highest in slow life histories. So although I'm now going to spend the rest of the next 5 to 10 minutes trying to convince you that density dependence matters a lot, at this very zoomed out scale, we do recover these past results of life history being related to these resilience metrics. So now I'm trying to think about how the resilience metrics change within a life history strategy when calculated at different population densities. So it's a little bit confusing, but the true model is density dependent. Our actual population that we are thinking of is density dependent, but the resilience metrics are only defined for a density independent model. So we say, okay, what if we had observed the population at low density? What would we think its resilience should be? What if we had observed the population at high density? What would we think its resilience should be? And I can calculate these at a whole range of population densities, remember from 0 to 1, meaning far from carrying capacity, and 1 meaning at carrying capacity, and then ask about the slope of these lines to understand how different life history strategies might be affected by this sort of ignored or neglected density dependence. Yeah, so then I can repeat for lots of life history strategies and the different density-dependent vital rate scenarios. So I'm going to show you now a series of color contour plots that summarize this, which show the slope of resilience against population density. So now the color contours are the slope of one of these lines. So the slope of compensation versus density. And then I found that the patterns were generally similar across different values of juvenile survival and retrogression. So I'm showing the results specifically for a juvenile survival of 0.6 and a retrogression rate of 0.3, but showing how it differs across different maturation rates and different adult survival. So that slow life histories are up in the upper left and fast life histories are in the lower right of these panels. So compensation responds to density most strongly when reproduction or juvenile survival are density dependent. So we have the darkest colors in this panel for when reproductive output is density dependent or when juvenile survival, but they're in opposite directions. So when reproductive output is density dependent, compensation always seems to be lower at high density. This makes sense because if reproductive output is lower, then your ability to boom and produce tons of seeds after a perturbation is going to be lower if you're already at high density. However, when juvenile survival is density dependent, then it looks like you are less likely to boom if you are at high density. And that you're more likely to boom. And that's actually a convoluted effect of a change in the population growth rate. So as you get closer to carrying capacity, the eigenvalue of your matrix, so your implied long-term population growth rate, is changing. It's decreasing. And then you take the same population model and divide it through by that value, and you get this counterintuitive result that the population is more resilient when it's close to its carrying capacity. So the point here really is that these go in opposite directions and that this is a little bit counterintuitive compared to the biological interpretation that we really want out of these models. Resistance does something kind of similar where the relationship between resistance and juvenile survival is quite direct. So when juvenile survival is density dependent, resistance decreases, you become less resistant as you're closer to your carrying capacity, but in the other cases, it looks like you become more resistant when you're close to carrying capacity. And it's the same kind of argument that the underlying model, the thing you're rescaling your model from, is changing at the same time as the things that you're actually interested in thinking about. So yeah, the point here is that it's not directly biologically interpretable, some of these patterns. And that we can't just say, density dependence might be biasing our resilience metrics, but in a consistent and predictable way. It actually matters which vital rate is density dependent to understand which way the bias might be going if we're ignoring this underlying density dependence. So the density dependent and density independent models also have distinct behavior. So this is what we call the transient envelope, which is similar. I showed you cartoons that were much smoother and prettier earlier, but this is an actual simulation of one of my density dependent models. And if we start them both from a population size of 1. So both models start from the same post-disturbance state. But the vital rates of the density dependent model, the orange dashed line, continue to change over time. And eventually, the density dependent model converges to its carrying capacity, while the density independent model converges back to lambda at some different population size. So, when we try to understand how different these two perspectives are in terms of recovery time, we see here that the density independent recovery time is 6 time steps and density dependent is 12. Obviously, it's important that these are recovering two different things. So the question we're asking is obviously different. But if we just want to know, do they go in the same direction? Do they predict the same, do they both predict longer times in a consistent way? What we find is that the density independent model consistently underpredicts the recovery time of the density dependent model. So here, this is the density independent model's recovery time and the density dependent model's recovery time and the one-to-one line, and we're consistently above it. But we don't see a big pattern in the offset across our life history space as defined by the vital rate PCA. So we wanted to check whether it was related to any other life history traits that we could pull out of our models, like generation time, which is typically our best measure of slow life histories and has been shown in the past to be related to things like damping ratio and recovery times. We expect really long-lived species with slow life histories to take a longer time to recover from perturbations. So what we find is that this offset, the amount by which the density-independent model is wrong about recovery time, is also related to generation time and to iteroparity. So the density-independent model does a worse job as generation time increases. Okay, so found that resilience metrics either increased or decreased with density, depending on the vital rate target of density dependence, and that the density-independent model consistently underpredicted recovery time of the density-dependent model, and that underprediction increased with generation time and iteroparity, which is bad news for trying to conserve the large herbivores that we have such a hard time conserving already. But further research is definitely needed to understand how we can analyze and quantify resilience in density-dependent populations. And so that's what I'm trying to work on, both in this project I'm going to tell you about and in my time here in Seville. So I've been working on thinking about how we can do density-dependent population recovery projections for data-poor species. And this project came out of, I was just contacted somewhat randomly by Darren Norris, who's a researcher at Amapá, at the Federal University at Amapá in Brazil. And he works on these very cute turtles, the yellow-spotted Amazonian river turtle. And he had been working on thinking about doing an IUCN green status evaluation to understand how halting hunting pressures on these turtles could be a good news story. So the IUCN green status list is to try to track conservation successes and species where we have managed to improve their level of risk of extinction. But Darren found that when he used his models, if there's a high level of harvest, then you predict that the population will go extinct, like this yellow line. If you have none, you predict that the population is going to explode. He tells me that a reasonable number of females for the sort of area represented by his model is 10 to 12 adult females. And this predicts that we're going to have like 500 of them per, whatever, per hectare or whatever area his model represents. So these are super unrealistic, and he felt that it would really help to be able to do these projections with density-dependent functions. But to make that accessible to conservationists and IUCN species experts, we need flexible tools that don't require people to build extremely complicated models. There's other considerations for projecting density-dependent population recovery and reasons that people maybe aren't doing it. Often the species that we most want to conserve are data poor. There are few individuals. If we really want to recover the populations, then the current population size might not be that big. Along with that, the populations we want to restore are at low density, where the negative density-dependent factors are going to tend to be weak. So being able to actually predict or fit a statistical model to what will happen at high density isn't something we might know. And we might not get the shape of that relationship very accurate because we only have data at the low part of that curve where there's a very weak relationship or only a linear relationship. But we may have an idea of how the density dependence should operate. That might come from biological and ecological expert knowledge. For example, we might know that this species needs a certain amount of space for its nest, and therefore this breeding area can only have X number of nests, and that sets a limit. That gives us some information we can use to put density dependence back into these models. Also, sometimes we might be able to borrow information from closely related species. but we still might not know the exact shape or strength of density dependence. So the solution that I've been working on is to build some generalized tools for exploring and comparing different scenarios. So the idea here is not that we're doing like really good statistics. The idea here is that we're using the simplest model we can in a user-friendly format so that you could say, here's my starting model. What if there was density dependence on this vital rate, and what if it looked like this? What if it's logistic? What if it's Ricker? What if it's Beverton-Holt? And be able to compare what those look like and what it predicts about your population carrying capacity or long-term projections. So I've built a Shiny app called Shiny Pop Recovery that's available online, and I'm going to beg you several times in the next few slides to go to it and tell me all the ways it's broken, because the only way that we figure out how to make good software is by letting lots of people break it. And then the other thing I'm working on is an R package, which I'm calling Denser Pop, which is still in development, but the Shiny app is going to use all of the functionality from that package so that users can use the Shiny app as a user interface, a really friendly user interface. But if you wanted to dig into more things, to make your own figures, you can then use the same package with the same functions as the app, but on your desktop in RStudio. Okay, so I've developed, not revolutionary, but trying to make the simplest and most generalized approach to density-dependent matrix population models. So a given vital rate of interest at a given population density is that rate at a population density of 0 multiplied by some scaling factor. Oh, sorry, this was animated. So this is the vital rate at low population density. We multiply it by this density-dependent scaling factor at a given density, which gives us that vital rate at a density n. Yeah, and our population density can be total, or it can be stage-specific, or it can be any kind of weighted function of your population. You could calculate the biomass. You could do whatever way you want to calculate that density. And then there are different ways that you can apply this scaling factor to your population of interest. You could apply it to the entire matrix and say that every vital rate will be decreased by density with the same strength. You could apply it only to the fertility entries, or only to the survival entries, or to particular sets of matrix elements. So you could say it only affects the adults the first time they're establishing a nest, which is actually one of the examples. Yeah, and maybe you have several matrix elements. Maybe you want all of the elements that indicate growth, or all of the elements indicating progression to the breeding class. Just depends on your exact model. We then also have diverse functional forms that this can take. So this is, what is that scaling factor? What's the shape of that scaling factor? These are the ones that are currently in the app. If you have others that are your favorite that you think are really relevant across lots of populations, I would love to hear them. So the logistic is linear and leads to a carrying sort of a single value carrying capacity. The Ricker model is that negative exponential that I used in the first project that I told you about. Beverton-Holt is somewhat similar to Ricker, but is a bit weaker. So it doesn't drop as steeply. And then the theta logistic is another one that has been suggested. It still drives the population to a carrying capacity, but it can have different shapes so that it doesn't assume a linear response along the way to the carrying capacity. Yeah, and then we can have all different strengths depending on the parameters of each of these functional forms. So a couple of examples of how you would actually apply this. This is how you might apply the Ricker model to the whole matrix. So you have the Ricker model equation, and you just multiply your matrix model that you've measured for your population at low density, or what you assume those vital rates would be at sort of 0 density. And then if we wanted to apply it to survival only, we would separate out the fertility matrix or fertility elements, and then apply that Ricker model only to the survival elements. So I've built this Shiny app Shiny Pop Recovery, which is an interactive web app. It's super fun. If you haven't built a Shiny app before because you think it's intimidating, it's actually really cool, and I was impressed with how quickly you can get one up and running. So yeah, it's been pretty fun to think about that side of things. I have a series of example species that are already loaded into this app. The white-tailed deer and gray wolves are both examples from Michigan, where the populations were observed across different densities and were sort of introduced. Well, in the white deer, they were introduced into an enclosure and then tracked as the population reached its carrying capacity in that enclosure. And then the gray wolves are from census data from the state of Michigan, where wolves had been locally extirpated in Michigan and then recolonized from further west in the US, from Wisconsin. And then the saltwater crocodile is a very complicated model that pushes the limits of what my generalized functions can do, and that's why it's a useful example. That's a model from Australia where they had really detailed information, and there are different density-dependent processes affecting different groups with different, you have to sum over different classes for each of the different density-dependent functions. It's a very complicated model. The peregrine falcon is quite a cool one because the density dependence operates on the probability of acquiring a breeding territory, which, as I've mentioned, I think is a really realistic and interesting place to put density dependence on populations we might not know that much about. And then the Ogawa Forest Reserve is mixed species, density dependence affecting, I think, only reproductive output. So as a user, when you approach the app, you get a panel that looks like this with lots of options. You get to choose the species that you might want to work with. Eventually, I would like it to be able to pull any model from the Comadre and Compadre databases, and also allow you to put in your own matrix in the app. You choose the functional form of density dependence. You set the parameters. So in this case, it's the logistic function. So the parameter it gives you is, it asks you for is k, but the other functions have different parameters, and this dynamically changes so that you only have to put in the parameters that are relevant to the function you've chosen. You choose how to apply it, which is what I was talking about, the whole matrix, or only survival, only fertility. Which stages should count towards density, how long you want to project, and then you also get to choose the initial population vector, but in my playing around with it, that doesn't matter so much. But if you want to start from the population you observed right now, you can also input a population vector. Yeah, so it ends up looking like this. There's information on the quick start guide. This plots the actual simulation. This panel tells you what your functional form looks like that you chose, and then the matrix panel is background information and metadata from the published study. Oh yeah, this is the density dependence panel. So in the case of logistic density dependence, you get a linear relationship like that. Yeah, so I would really love it if you would play with this app. And I'm being a bit flippant saying break it for me, but actually it's so useful to see people's feedback and to be told what's not clear, what's your impression when you arrive at the website, what information do you need, and what capabilities do you wish it had, and also what species or model do you have a citation for that you think I should add, because having good examples is super, super valuable. So I just wanted to end on what am I doing here? So I'm doing a research stay with Maria Paniw and team until December 12th. And yeah, my Marie Curie fellowship is focused on density dependence, but I have pretty wide interests in methods development and population dynamics in general. So I'm hoping to work on more complex models than what I showed you today. That 2x2 model is way too simple for a lot of species. So some possibilities include fitting density-dependent structured population models to reanalyze existing demographic data sets. We've talked about marmots or Cryptantha flava. And then also thinking about extending some of this resilience research to think about how we can do fair comparisons across taxa with very different life cycles. So one of the things that's really missing in the databases that have been used for a bunch of this macroecology, particularly coming out of Rob Salguero-Gomez's group at Oxford, is that there are, at least on the animal side, it's really, really concentrated in mammals and birds. And there are almost no invertebrates. And, you know, in trying to think about why that is, one of the clues is that a couple of the matrix models that have been built for invertebrates have issues, like they don't fit very well, or people have struggled to get the insect life cycle to actually match with a matrix population model, and people sometimes make mistakes or just like can't get a good statistical fit with those kinds of models. So I've been talking to Maria about some of the other ways, right? There are lots of other ways people could model invertebrate populations, and if we want to be able to do this kind of synthesis macroecological study, then we need to know how we can compare these different taxa. Even if we can, maybe trying to model insects with a matrix population model is a fool's errand, and we should not do that. If we shouldn't do that, then we need to think about how we can make comparisons across model types. So that's one of the things that could be really cool to try to work on. Then the last thing I wanted to tell you is that in January, I'm starting a tenure-track faculty position at Old Dominion University in the USA. It's here, this is Chesapeake Bay and Virginia Beach, and the university is very close to that. So I'm going to continue to work on quantitative biology and also do field work about marine and coastal population ecology. So I would love to talk to anybody who's interested in collaborations or just having interesting conversations. Yeah, one more plug, please play with the app. And then also email me or come find me. I'm over in the other building in office 1-7. And this is my email address into the future. I would love to talk about any other possible collaborations on anything related to population dynamics. And then my PhD was on fish, larval fish, and I would love to talk about fish. It's something I haven't gotten to work on very much in the past 5 years, and I'm really excited to get back into it. So yeah, if anybody here is doing any aquatic kind of research, I would love to talk about it, but probably not that much, because if you're more focused on Doñana. Anyway, yeah, okay, thanks.
Cool, thank you very much, Christina, for your talk. And we have time for questions.
Hey, Chrissy, great talk. I really enjoyed it. So I have a question, and it might be something more for what you're working towards the future. In the first part of your talk, from what I understand, if I have a species that I work with and I know it's density dependent, basically until I actually know those relationships, I shouldn't touch any of these resilient matrices. Or are there specific life histories or specific components of the model that you're like, well, if you know this, then you can go ahead and give it a try?
Well, I think if your population is at a given density, and that seems to be mostly constant, then you can ask what the resilience of that population is sort of right now in these metrics. Like, what's going to happen? It definitely does a good job of what's going to happen in the first time step. If there's a perturbation, what's going to happen in the first time step is just the stuff you collected now, right? But if you're trying to make a projection 100 years into the future, it's not going to do a great job, and it might not do a great job of estimating recovery time. And if we're trying to use these tools to, I mean, you could also argue that some of the issues are in the direction we would already understand. So if the bias in recovery time is that long-lived species are even slower to recover than we thought, then maybe that's okay, because we still know they're going to take a really long time to recover, and maybe it depends how true you think your model results are, right? How much stock do you put in the population growth rate that you get out of a matrix model?
That makes sense. Thank you.
I'll just go. Sorry. Yes. Yeah. So thank you very much. It was really lovely that you managed to do such a theoretical talk with really nice examples and explaining it quite well. And so one thing that I was always wondering, and this is opening probably a whole Pandora's box, is what about the environment? Because we talk about K, but really what's going on in most populations is the interaction between actual environment and density. Yeah. So, like for instance, you say, what is your population at low density? But I would say, but it matters what is the environment. Is my population at low density because it experienced density dependence, or how's the environment now, basically,
Yeah.
So that's sort of like, how is it even possible to, because the environment is also such a broad thing.
Yeah.
Yeah.
No, I think these are really interesting questions, and I'm excited to talk more about like the forecasting and the digital twin kind of work, which is really trying to make very complicated models that try to take into account a lot of information, right? Whereas I tend to come from this more theoretical side of things, where I'm less concerned with whether I'm forecasting what's happening next year or in 2 years, and more interested in these big patterns, in which case I want the simplest possible model so that I can point to exactly which thing is causing the pattern I see. So I think I think there's a lot of really interesting work that's in that, like, how do we talk to each other, and how do we reconcile these like theoretical insights that come from far too simple models and what we might want to actually do when we're trying to make predictions and do forecasting. And some people would say, well, if you're interested in life history evolution and you want to think on long timescales, then maybe you need a stochastic model, but mostly you're thinking about what the mean environment is doing to you, or something like that. And so knowing exactly what the environment is right now might not be so important. And so, I don't know, it's like it all comes back to like, what's the question you're trying to ask?
Yep. Okay. Thank you.
Well, thank you for your seminar. Well, congrats for your AMNAT and your tenure-track. You are on fire. So congratulations. So I have two questions. The first one is related to Maria's, actually. So how have you dealt with stochastic variation in vital rates? Because I have the feeling that the stochasticity overrides density-dependent effects in nature.
Yeah, that might be totally true. I'm not doing stochasticity in the first paper, where I'm asking how does density dependence impact resilience. There's no stochasticity. We're just saying we have deterministic density dependence. So absolutely, that's important. It's not in that study. In the Shiny app, I think it would be really nice to be able to include some stochasticity into those projections and allow people to give some kind of distribution of vital rates or something. So, and then you maybe have to think about how to disentangle. Yeah, if the stochasticity is strong enough, then like either the stochasticity swamps the density dependence, or they have some kind of interesting interaction. So yeah, I think that's a really good point, and I would like to add it to the Shiny app, but it's not in the AMNAT paper. Yeah.
But the tools exist, so it's something that you can just put in there. Anyway,
yeah.
Basically because stochasticity allows you to get far from these unrealistic parameters like equilibrium, carrying capacity, these kind of things that they exist in our minds, but in nature is different.
Yeah.
My second question was related to the Shiny app, actually, and you said that the user can choose whether density dependent affects one vital rate or two, or I find this quite dangerous.
Okay.
Because what about trait correlations?
Yeah.
So perhaps the user should choose the kind of relationships among vital rates. Because you can model one vital rate not related to the other, but we know that that's not what's happening.
Yeah.
Right.
Yeah.
So I think that the user perhaps could use the kind of relationship between vital rates, in these 2x2 models, because I think the Shiny app also reduces the models to a 2x2. No.
No.
Okay.
The Shiny app allows you to use whatever
I
size
see.
model you have.
I
Yep.
see.
Yep.
But the
it's a really
relationship
interesting
between
point.
traits. So you could offer independent variation.
Yep.
More mates, more reproduction, these kind of things.
Yeah, absolutely. Yeah, that's a really interesting point. Yeah, thank you.
Thank you.
Any more questions?
Can you test your models to see whether the density-dependent models predicts population dynamics better than density-independent models?
Well, yes. If you have a time series, then you totally can do that. But the part of the idea with this Shiny app is like, what do you do if you don't have that time series? If you can't do the statistical analysis and you're just going based on sort of expert knowledge, how do you proceed with trying to make any kind of prediction? But yeah, absolutely, that is very important. But this is a scenario testing kind of approach where we say, we don't know how it should function, but we kind of have an idea of what the population size should be, and we kind of have an idea of which vital rates should be affected. And so we just want to try to think about what the outcomes might be. There's really...
The outcome of the models depend on the carrying capacity of the population? Or you assume that all population have the same carrying capacity?
Sorry, for the Shiny app?
No, from the first part of your talk?
Oh, for the first part, yes. Sorry.
Because you showed that the carrying capacity was like 1. I guess that's like a normalization?
Yes.
So you are assuming that the recovery time and resilience or do not depend on the carrying capacity of the population?
Right, because those metrics are calculated only from a matrix. They don't, the current metrics that exist don't have a way for us to take into account carrying capacity. So that study is completely theoretical. I'm making everything up. I don't have populations to compare to and ask which model does a better job. I'm asking if the true dynamics were density dependent in a way that I've just picked, how do these other metrics we calculate fall short of biological interpretation?
But I'm thinking that you said that we should have a time series or time series data to test the prediction of the models.
Yeah.
And I'm thinking that perhaps you only need the carrying capacity
Oh,
of those
I
time
see.
series, right?
I see. Mm-hmm.
So if you know the carrying capacity, could you check whether density-dependent models predict better than density-independent
Well, they almost
models?
certainly will, because a density-independent model, I mean, I guess it depends on the time scale that you're most interested in, but if you just want to know whether your population will go to its carrying capacity, a density-independent model won't. It will grow exponentially or shrink exponentially forever. Yeah, yeah.
There was another.
Hello, thank you for your talk. It was very nice. I have a curiosity, and it's kind of in line with Maria. I'm not theoretical at all, so I follow your talk as I could.
Yeah.
But I work on seed dispersal,
Uh-huh.
frugivore-mediated seed dispersal, and there are some new research that it seems that frugivore could make some kind of negative density dependent on fleshy fruited communities.
Mm-hmm.
So I'm very curious about if these kind of models could be like adapt to a community, considering like plants and animals.
Yeah.
Because it's very nice. They also see like variation along the seasonal, along the fruiting period.
Yeah, yeah. No, that's a really cool question. And actually, part of the reason that I think density dependence is something we have to work on in population models is because that is a crucial way to connect us up to community models. Community models are always sort of frequency dependent, or they have interaction coefficients, right? So you have the interaction between different species, which already gets you some model that is nonlinear, because it depends on how many of species A and how many of species B do you have. And so, yeah, I think that's like a really important connection to be able to make. And there are, I think, nowadays more and more efforts to bridge the population view and the community view. And I think that these nonlinearities, like density dependence, are crucial for making those connections. So I don't have an answer of whether I'm doing that, but yes, that is exactly where I would want your mind to go. It's good. Yeah.
Thank you for the talk. Does the model include positive density dependence?
Not yet.
Not yet.
Yeah. Tell me about positive density dependence that matters.
Yeah, alley effects.
Yeah.
For instance.
Yeah. Do you work on a system where that's important?
Yeah.
What is your system?
When you have a small population, as you say,
yeah,
that is more likely to have positive density dependence than negative density dependence. So,
absolutely.
So,
I just need a good example model or a good example species where I can play with that and have something that gives me sort of motivation to add the functionality.
For example, you have a reproduction between plants. If you have very few individuals, they don't mate because they don't find each other because there are so few of them.
Yeah.
So they need more plants to find each other and to reproduce.
Yeah, absolutely. Yeah. This was mostly just trying to keep it as simple as I could, but I think that is a really important thing to include as well. I totally agree.
Okay.
Yeah.
Thank you.
Yeah.
Just out of curiosity, sort of going back, there aren't any standardized models that do sort of hump-shaped density independence? Because it's that the Ricker and Barrett and Holt, they all go down. So there's nothing that goes like this?
No.
Really?
There's a book on alley effects. I think I forget his first name, but Corchamp is the one of the main authors that has written about alley effects. And somewhere I remember seeing this table of all different functional forms that have been used, but there isn't a canonical functional form, which is part of the reason that I haven't put it in, because you have to pick something. And typically the alley effect models then have a Ricker behavior or Beverton-Holt behavior later. So it's sort of a modified.
Just that in the beginning would go up, and then it's just basically a U shape.
But you still have to choose. You have to choose so many things about that curve. So I think that's why there isn't. It's in the It's in my example.
Yes, that's the flexible one, right?
It's in here from my proposal. But I should look back at what function did I use to make this cartoon for my proposal, and then put that back into the app. Yeah.
The first one. The first one on the right, on the left.
Yeah. Exactly. Yeah. Okay. Yeah. So I should just put that back in the code.
Actually, relating to functional forms, I was curious, why didn't you choose a sigmoid? Because something that I usually do, because they have two saturation points, one in both extremes of the density, so maybe that could be also useful.
That's great to know. Yeah, I was kind of just building up from examples that are in structured models. So this is what I want to hear, what function makes sense in the systems that people actually work on, empirical systems. So great. I'll look for a sigmoid function and add that. Yeah.
Okay, Cristina will be here until December, so you
Yeah.
have plenty of time to ask her more questions. And please let's send her again, and see you next week.